Greetings, Brookings Bio students. This is Mrs. Rydell, and in this chapter we're going to take a look at how living things use the sugar that was produced in our last chapter when plants do photosynthesis. And we're going to look at something called cellular respiration. So if you remember from our last chapter, we talked about autotrophs, the ability to make your own food. And autotrophs use energy from sunlight or chemicals to make their own food. And we talked about producers in a couple different chapters so far. In our last chapter on photosynthesis, green plants were using photosynthesis to catch sunlight, energy from the sun, and turn it into food molecules, into glucose. And in this chapter now, we're going to look at what's the next step? What happens to that glucose after it's produced? We're going to take the glucose now and break it down and use the energy that's released when the molecule of glucose is broken apart to provide the power to charge up our ATPs. We talked about ATP before. In the process of doing that, breaking down sugar and using that energy that's released is called cellular respiration. And what kind of organisms do cellular respiration? All living things do respiration. Plants do it, fungi do it, animals do it, and that includes you, humans, our animals. Bacteria do it. All living things need energy. And so they can't all make their own food, but they all can use food to provide the power that they need to do cellular processes. How do we get the sugar from the plants? Remember, we are heterotrophs, and heterotrophs are organisms that get their energy by food molecules that they get from eating other organisms. So we get the energy that the plants stored as glucose when we eat plants directly, or when we eat animals that ate the plants. So when we're taking in those food molecules, then we're getting the energy that's stored in that food, and we can use that energy to charge up our ATP and to do all the activities that cells need, bodies need to stay alive. So let's dust off our bio brain a little bit and remember what we have talked about in some previous chapters. Make some connections to things you already know. Remember, we did a whole unit on cell parts. So see if you can remember the area with the gel-like goo material inside the cell membrane surrounding the mitochondria and all the other organelles is called cytoplasm. And some of respiration, the pathway of breaking down the sugar that we're going to see in this chapter is going to happen in the cytoplasm. We also learned about this power plant organelle. This power plant of the cell burns glucose and stores the energy as ATP. That was what we learned in our cell parts chapter. Now we're going to take a look at how that actually works. How do we burn the sugar and capture that energy? And the cell part that's involved in doing that is our mitochondria. Okay, so let's take a little peek here at our mitochondria. And some of the parts of mitochondria we learned back in our cell parts chapter. But some parts are, we're gonna add some new names to things. So remember, we learned that mitochondria and chloroplasts and the nucleus are all organelles that have a double membrane. It's one of the few organelles with a double membrane. So we have an outer membrane and an inner membrane. So we're going to do a little coloring here. And I'm going to kind of color code this because my outer membrane here is going to be this part of the mitochondria. It makes kind of a container here. And remember, mitochondria, more than one. Mitochondrion is a single. Usually we kind of just talk about mitochondria in general. Okay, so I'm going to color this outside layer. That's the outer membrane. If you don't have this color, you can color whatever color you like. But we're going to kind of color code this. So label your mark your label with the same color that you're using to color the part. And then we also learned that there's an inside membrane. And remember when we colored our, or when we talked about mitochondria before, we said that cells have an inner membrane. Sounds kind of like a girl's name. And that's the cristae membrane. We learned that back in our cell course chapter. So I'm going to color this, and remember it's kind of like a folded, looks like a folded ribbon in the picture here. And if we follow the ribbon around, kind of just pick a place and start following the path, and we follow it all the way around inside the cell, what you notice is it starts to make compartments. The folded cristae divide the inside of our mitochondria into two compartments, because there's going to be a place that's between the cristae and the outside membrane, and then there's also a place that's inside the cristae membrane. So we're making compartments. One of the things that making membranes allows a cell to do is to compartmentalize things, to make little sections where different things can happen in different places, and that's what the mitochondria are doing. They're going to allow different kinds of chemistry to happen in different places because of that cristae, and folding it gives us more surface area for chemical reactions to happen. So cristae membrane, in whatever color you colored your cristae, let's kind of mark that here, circle it somehow, so we know that that's our cristae. And now if you look at the picture, we kind of have two spaces now. There's a space between the inner membrane and the outer membrane, and that is named intermembrane space. That's kind of logical, intermembrane space. If you think about that word inter means between, like interstate highways go between states. So this is the place that's between the two membranes. We call it the intermembrane space. So let's fill that in. And then we're going to kind of color code this, so we'll mark our label with the same color that we use to color that intermembrane space. And you're going to see when we start talking about respiration, some things are going to happen here in that intermembrane space that are different than our other space. Okay, I think we got that colored in. And let's mark our label here. Intermembrane space is the space between the inner and the outer membrane. And then the other space that we have is the space that's in the center, the matrix. Inside my cristae membrane, there's also a little compartment here. It sits in the center in the folds of the cristae. And we're going to see some different things happen here during cellular respiration too. So we got to kind of have some name labels to put on places when we talk about what's happening there. Okay, so matrix is my space that's inside the folded cristae. And then remember, this is inside a cell now. This is a cell part that's floating inside that cytoplasm. My cytoplasm now is going to be this area outside the mitochondria, and the mitochondria is a cell part. It's going to be floating in the cell, and cell contents are floating in that kind of cytoplasmic goo. Okay, I think we kind of have some locations now, and we can talk a little bit about this whole process. So all organisms, heterotrophs and autotrophs, are going to use the energy that comes from burning their food to charge up their ATPs. And remember, we talked about that before in previous chapters, that we can store energy by making that high energy bond by adding the P, and then releasing the P releases the energy. That phosphate group breaking that bond releases the power, and then I use the energy from food to charge that up again, make a new bond, and then I can release the power. We kind of compared it to a rechargeable cell phone battery, and that's our rechargeable power molecule in all living things. Doesn't matter if you're a bacteria, a worm, a human, a monkey, a tree, fungus, all living things use ATP as their power molecule. And we can charge it and uncharge it, charge it and uncharge it over and over and over again. And we talked about that in past chapters. This process of releasing the energy from food, we're burning glucose now, to make ATP is called cellular respiration. And that's what this chapter is about. What are the steps that a cell goes through to take the energy that's stored in glucose and release it slowly to capture that energy and charge up our power molecules, charge up our ATP. If we did this quickly, most of that power would be lost. If you think about a chemical reaction like an explosion, the power comes off of that pretty fast. And it would be very difficult for the cell to capture that energy. So respiration is going to happen very slowly, lots of little baby steps. Remember when we did photosynthesis in our last chapter, we didn't just take carbon dioxide and water and stick them together and bam, we got sugar. Lots of little baby steps to get to that point. And we're going to do the same thing. We don't want all of the energy to be released all at one time. It would be too hard to capture that. So if we did that, most would be lost as light and heat. And we wouldn't be able to capture the energy that's coming off. So we're going to release that power, release the energy from glucose a little at a time. Little baby steps. Let's go back and review a little bit, because remember when we talked about our last chapter, we had to, I asked you to learn the chemical equation for photosynthesis. We took 6 carbon dioxides and 6 water molecules, and we used the energy from sunlight to make sugar. C6H12O6 is a glucose molecule. It's a kind of sugar, a carbohydrate. And that process released oxygen. Remember, plants in that photosynthesis process give off oxygen to the atmosphere, 6 O2s. I'm going to put the equation here for respiration, and then I want you to kind of look at the two and see how they're similar. So in respiration, we're going to take the sugar now that we made during photosynthesis, the plants made, C6H12O6. And respiration, when we think of respiration, think of breathing. The gas that we use to do cellular respiration is the one that we take out of the atmosphere when we breathe, and that's oxygen. So I'm going to add 6 oxygen molecules. And we're going to do cellular respiration. And respiration produces 6 carbon dioxides, 6 water molecules, and the energy now that's going to be released, we're going to use to charge up our ATPs. If you take a look at those two equations, what you notice is not only do they have the same molecules in them, but they're exact opposites of each other. The things that we are making in photosynthesis, the products, are going to be the reactants in our cellular respiration equation. And the reactants in our photosynthesis equation are the products of respiration. They're the exact opposites of each other. What cellular respiration does is to undo the process that we did in photosynthesis. In photosynthesis, we put the energy into our food molecules, and now we're going to take those food molecules and get the energy back. So when we talk about food molecules and energy, we need some vocab here. And in science, the amount of heat energy it takes to raise 1 gram of water 1 degree Celsius is called a calorie. Notice it's written with a little c, lowercase c, calorie. If we were to measure the energy in food, we need a little bigger unit, because the amount of heat it takes to raise 1 gram of water is not very much. And food molecules have a lot more energy than that. So we're going to use a unit to measure energy in food called a calorie. And notice both words, calorie, calories, kind of the same. The difference is capital C. 1 capital C calorie is the same amount of energy as 1,000 little calories. What that means is 1 calorie in food, a capital C calorie, is the amount of heat energy it would take to raise 1,000 grams of water 1 degree, or 1 gram of water 1,000 degrees. There's a lot more energy in food, and we can see calories when we read food labels. It'll tell you how many calories are in a serving of this food or that food. And it's a measure of heat energy, how much energy is stored in that food. And we actually measure calories by taking a sample of that food and burning it in a special device called a calorimeter, and allows the scientist to measure how much heat is coming off of that food when it burns, and raises the temperature of the water, and that's how they decide how many calories are in the food. Okay, in photosynthesis, when we talked about photosynthesis, we use this electron carrier, NADP+, to carry our high energy electrons. And respiration is going to use electron carriers again to transport electrons, but we're going to use a couple different ones. And they are called NAD, NAD+, and FAD, FAD. Notice you got an NAD, which is very similar to the NADP. How are we going to remember that the NADP+ goes with photosynthesis and the NAD goes with respiration? I always remember the P in NADP kind of reminds me of the P that starts photosynthesis. The word photosynthesis starts with P, and the carrier that we use to do photosynthesis is NADP+. So we're going to use these NAD and FAD carriers, and we're going to kind of charge them up the same way we did before. We wrote equations to show how we charged up NADP+ to NADPH, and it kind of works the same way. I'm going to take my NAD+, and I'm going to add electrons, 2 electrons and a hydrogen. Remember, we're adding the hydrogen to balance out that negative charge, a plus on our NAD and 2 minuses on electrons. That plus on our hydrogen ion is going to balance that out. So before we did NADP+ and we charged it up to NADPH, we're kind of going to do the same thing now. NAD+ is going to charge up to NADH. And that's the charged up form. That's the form that's carrying those electrons. FAD is going to kind of work the same way. We're going to take FAD and we're going to add 2 electrons. But this time now, notice the FAD doesn't have a charge on it. So when I add 2 electrons, 2 minuses, I also have to add 2 pluses to make that balance out. So we're going to take 2 electrons and 2 hydrogen atoms. And if I add 2 hydrogens to FAD, I get FADH2. That's the charged up version of FAD. And both of those are going to be our electron carriers now to carry electrons inside of our cell and to help us with this cellular respiration process. Okay, so the first step in respiration is called glycolysis. And glycolysis is going to happen even before we get into the mitochondria. It's going to happen outside the mitochondria in the cytoplasm. And so let's talk about that word a little bit. Glycos in Latin means sweet. And lysis, we had lysis before. We had hydrolysis, breaking down, a chemical reaction to break down things. We had lysosomes before. Plasmolysis, lysis means to split something apart. So the word glycolysis is kind of telling us what is going to happen in this reaction. We're going to split apart the sweet. And the sweet is glucose. Glycolysis is going to split the glucose apart, split it in half. And this process does not require oxygen. It does require a little bit of energy to get it going, kind of like a little kick in the pants to get it started. So if we need energy to get this going, what molecule do you think is going to supply the energy to get glycolysis started? Remember, we talked about energy molecules in cells, and our energy molecule, our rechargeable cell phone battery, is ATP. ATP is going to provide the power to get this whole process going. And that seems kind of backwards, because the whole purpose of doing this is to get some energy out of the food. But we need a little ATP to get it started, and you'll see that we're going to get way more back in the end. Okay, so let's write the equation for this. This is glycolysis now. We're going to start with glucose. And glucose is a 6-carbon sugar. And so we have 6 little C's here, 6 carbons. Remember, glucose has oxygens and hydrogens as well, C6H12O6. But those oxygens and hydrogens are attached to the carbon part of the molecule. So if I follow where the carbons are going, I also can tell where the oxygens and hydrogens are going. So we're going to just follow the carbons. So 6 little carbons in glucose. And we're going to add 2 ATPs to get this thing going. And basically, we're going to do some chemistry, hocus pocus. There's actually about 10 steps to this process. Remember, little baby steps. Doesn't happen all at once. But the result of that reaction is I'm going to get 2 molecules that each have 3 carbons in them. Basically, I'm taking my glucose and splitting it in half. And I get a molecule called pyruvic acid. Okay, let's add this word on top. Pyruvic acid. Pyruvate. Sometimes you will see it referred to as pyruvate, and for our purposes, those 2 words kind of mean the same thing. Pyruvate, pyruvic acid. It's a molecule that's got 3 carbons in it. And by splitting my glucose now in half, I get 2 pyruvates, 2 pyruvic acids. And that process, anytime you break bonds now and do some chemistry here, it's going to release some power. So I'm going to get, so I put in 2 ATPs, but I got 4 back. That's kind of a good deal. Remember we said we got to put in a little to get it started, but we get more back during the process. So I kind of gained net gain of 2 ATPs. If I asked you if you give me $2 and I'll give you 4 back, would you go for that deal? That's a pretty good deal. You doubled your investment, and so even though I had to put a little ATP in to get it going, I'm going to get more ATPs back. So net gain of 2 ATPs and 2 NADHs during the glycolysis part of the reaction. Now the cell has a choice. If there's no oxygen, we call that anaerobic. An means not, and aerobic has to do with air. Think about aerobic respiration. If there's no oxygen available, the cell is going to follow the pathway, the next step in the pathway, into a process called fermentation. So here's my pyruvic acids that I made from splitting my glucose. And without oxygen, it's going to follow this pathway into fermentation. If there is oxygen available, we call that aerobic pathway with air. If there's oxygen available to the cell, the pyruvic acids are going to follow the with oxygen pathway. And the next step will be to go into something called the Krebs cycle and electron transport chain. And kind of is determined by the environment that the cell is living in. So if there's not enough oxygen available, the cell will do fermentation. If there is oxygen available, the cell will follow the with oxygen path and complete cellular respiration. Okay, so let's talk a little bit about the fermentation, the no oxygen, without oxygen pathway. And basically there's 2 kinds of fermentation, and the cell doesn't really get to pick. Each kind of cell can only do one kind of fermentation. There's alcoholic fermentation and lactic acid fermentation, and they're named for the products that are produced when the cell does fermentation. So what do you think the cell is making if it's doing alcoholic fermentation? It's making alcohol. And in lactic acid fermentation, the cell is going to make lactic acid. Cells can't switch back and forth. Each kind of cell does one particular kind of fermentation. So let's talk about how that works for those 2 kinds. An example of an organism that does alcoholic fermentation is yeast. Yeast is a eukaryote, and we use it in a lot of processes, used to make bread. So we're going to take those pyruvic acids that we made in glycolysis. And remember, this is the next step now after glycolysis if there's no oxygen available to the cell. So pyruvic acid, and we're going to use one of those electron carriers that we made, the NADH. And we're going to do some chemistry, and notice I'm going to go from a 3-carbon pyruvic acid molecule to a 2-carbon molecule. And that 2-carbon molecule is alcohol. Alcohol is the molecule that we find in alcoholic drinks. And to make alcohol from pyruvic acid, notice I have to release one of the carbons to go down to a 2-carbon molecule. And that carbon goes off as a gas. what gas do we know that has carbon in it? That's carbon dioxide. So one of the carbons is going to be removed from pyruvic acid here. We're going to release this carbon. And it's going to go off as carbon dioxide gas. And remaining 2 carbon molecule is alcohol. Now when we do this chemistry now and the NADH drops off its electrons, it's going to go back to being the uncharged form, and that goes back to being NAD+. So we're using some of the power that's in that NADH electron carrier to do the chemistry that we need to change pyruvic acid into alcohol. This happens when yeast makes bread dough rise, and the carbon dioxide bubbles that are released during this process are what make the little holes, the little air spaces in the bread. It makes your bread poof up. The alcohol that's being made, if you think about yeast doing alcoholic fermentation, when we eat a peanut butter sandwich or eat bread, we don't get drunk. And the reason is the alcohol that's produced in this process evaporates when we cook the bread. Another example of organisms that do alcoholic fermentation, taking pyruvic acid, using one of those electron carriers to make alcohol and carbon dioxide, and release that NAD+ carrier again, are the organisms that make alcoholic beverages for us. It happens when yeast makes beer. Happens when certain kinds of bacteria make wine for us, or spirits. The fizzy bubbles that are in the beer when you taste beer are coming from that carbon dioxide that's produced when the yeast does alcoholic fermentation. And the alcohol is the alcohol that's in our alcoholic beverages. Wine, they release the carbon dioxide out of the containers, before it's bottled, so that you don't get the fizz in wine. But champagne is a fizzy. Champagne keeps the bubbles in, the carbon dioxide in, and you have the fizzy bubbles. And these products that we drink are produced by that process of alcoholic fermentation, making the alcohol and carbon dioxide from pyruvic acid and releasing that electron carrier. Lactic acid fermentation is another kind of fermentation, and some cells do that. We're taking pyruvic acid again and one of those electron carriers, NADH, only this time notice we're not going to remove one of the carbons and release carbon dioxide. We're just going to rearrange the atoms a little into something different. And the something different is lactic acid. And just like in our last reaction, we're using the power from that electron carrier, the NADH, to do the chemistry. And that power, when it's released, puts our electron carrier back to its uncharged form, the NAD+. This happens in humans, muscles, during exercise. When you start breathing heavily and you're not getting enough oxygen to your tissues fast enough, your muscles switch into doing lactic acid fermentation instead of respiration. And buildup of that lactic acid in muscles is what causes the soreness after you exercise. Eventually your body can go back when the oxygen is available again and remove that lactic acid, break it back down again, and get rid of it. But muscle cells, when they're in a situation when they're not getting enough oxygen, are going to do lactic acid fermentation. Other kinds of organisms that do lactic acid fermentation, some kinds of bacteria use lactic acid fermentation to make certain kinds of foods and beverages that we like. We use those bacteria to ferment things like yogurt, buttermilk, sauerkraut, pickles, cheese, sour cream, kimchi is kind of a pickled cabbage. That kind of tart taste when you taste yogurt is coming from the lactic acid that's in the food, and the bacteria are doing that lactic acid fermentation on the sugars that are in those food products to produce the lactic acid that we taste in those kind of sour tart foods. So why do cells do fermentation? Why don't they just keep doing glycolysis and make their ATP from that? Because those products that we make during fermentation, the alcohol, the lactic acid, those are really poisons. They're not something the cells is wanting. And the reason that cells do that is that without oxygen, that pyruvic acid, if we just keep doing glycolysis here, eventually we're going to pile up a whole lot of pyruvic acid here. And all our NADH carriers are going to get charged up and full. And that means we can't continue the glycolysis pathway anymore. So without oxygen available to the cell, pyruvic acid builds up and all the NAD plus carriers get full, and that eventually causes glycolysis to stop. So we need something to do with the pyruvic acid that's building up, and we need a way to get those NAD plus carriers back. And that's why cells do fermentation. It's so that if you compare the equations in both of those instances, in alcoholic and lactic acid fermentation, what we're getting back at the end is those electron carriers. The NAD pluses come back when we're doing fermentation, and it allows the cell to keep doing glycolysis. Fermentation happens so cells can regenerate those NAD plus carriers. It's not that they want the lactic acid, it's not that they want the alcohol, it's that they want those NAD plus carriers back so they can keep glycolysis going. We oftentimes kind of have the misunderstanding that plants make food, plants do photosynthesis, and then animals do respiration. And plants do photosynthesis. They're making the food. But the reason they're making that food is so they can burn it when they need power. They're making their food so that they can do cellular respiration to get the energy back out of the food. So plants also need oxygen to do cellular respiration. They're not just doing photosynthesis, they're also using respiration to break those food molecules down that they built during photosynthesis. So if you think about South Dakota and make a connection to something that you know here, in the springtime, after farmers plant their fields, a lot of times our fields get flooded and they look like this, where we have standing water. And what happens if you have seeds there that have been planted and then the field gets flooded, those seeds, if they're underwater for more than a couple days, the seeds are going to die because the plant roots, the plants cannot do respiration on the food that's stored in those seeds to get them started and get up out of the ground so that they can start doing photosynthesis. But the beginning, they're doing respiration and having, think about you sitting underwater for a couple days. You're not going to have the oxygen that you need to do respiration, and plants are the same way. That's the end of our slideshow, and stay tuned for what's going to happen next when we follow the with oxygen pathway.